As we've mentioned before, we have the intracellular fluid inside of the cell, the extracellular fluid around the cells, and then we have a third environment, which is the interstitial fluid found in our tissues. Today's video is going to talk about the movement of molecules within these spaces. This video specifically is about passive transport. We'll have another video about active transport. Passive transport is called passive because it requires zero energy, and we are going to talk about a few different types: diffusion, osmosis, and facilitated diffusion. Diffusion is the random movement of molecules within a space, and the net movement is always going to go from a place of high concentration to a place of low concentration. That's not to say that there is no movement from molecules of low concentration to high, but there is way more movement from high concentration to low concentration. Molecules are constantly moving. An example of this would be like salt in salt water, or hot cocoa when you mix hot cocoa with hot water if you're making hot cocoa. There's always going to be movement of molecules. Another way to think of this is the oxygen molecules or nitrogen molecules that exist in our atmosphere. We're interested in diffusion through the plasma membrane. If you can remember, our plasma membrane is made out of that phospholipid bilayer. So it has phosphate heads and lipid tails, which means it has a hydrophilic area where the heads are, and then a hydrophobic area where the tails are. This impacts what can move in and out of the cell freely. Non-polar compounds can move through the cell membrane freely because of this phospholipid bilayer. So this includes carbon dioxide and water. And so what we see is that oxygen is going to move from an area of high concentration, like in this extracellular environment, to an area of lower concentration, as you can see by these blue molecules. Similarly, carbon dioxide is going to move to a place of high concentration within these cells to an area of lower concentration. This is literally what happens whenever we oxygenate our tissues. So the phospholipid bilayer is going to impact what can move through and what can move out. And one impact of that is that charged molecules can actually not go through this phospholipid bilayer so easily, not as easily as those nonpolar compounds. And so what they have to depend on is a protein. And these proteins can be ion channels, like these shown here, or they can be these kind of like swinging door type proteins. But if they have a helper, like these proteins to help them, this is called facilitated diffusion. It's called facilitated diffusion because they have a facilitator. They wouldn't be able to move through without these channel proteins. So a good example of this So in this image here on the right, you can see that there's this kind of balloon-like structure. This is supposed to mimic our cell membrane. And notice that certain things are able to move out or in, and certain things are not. Notice that these big purple proteins are unable to move out. And this is similar to the structure of our cell membrane. Very large proteins cannot move freely through the membrane. Notice these smaller ions, like the yellow ones, they can move freely through. And they're going to go from a place of high concentration, which is inside this cell, inside this dialysis membrane, to a place of low concentration, which is inside the beaker. Similar to the green molecule, which is supposed to delineate glucose, these are more already in equilibrium, And so we kind of have a zero net movement, which is what's shown with these like equal arrows. The rate of diffusion is going to depend on a few things. And you can think about real life examples that you have diffused molecules in solvent. For example, if you've ever made Gatorade from those like powder packets or hot chocolate from those powder packets, the rate of diffusion depends on the magnitude of the concentration gradient. And so if you have a really high magnitude area with a really low or zero area of concentration, there's going to be a faster rate of diffusion. It's going to depend on the permeability of the membrane. If this membrane is super porous and just allows anything through, it's going to diffuse really quickly. If it's not, and it's actually kind of tight and there's only really small areas that solutes can move through, it's going to change that. It's going to decrease that. Another thing that can increase or decrease the rate of diffusion is temperature, and maybe you've experienced this. If you're trying to make hot cocoa with hot water versus with cold water, it's easier with hot water. And another factor is surface area. And a good clinical example of this is your lung tissue. Your lung tissue, if you can remember from anatomy, they kind of look like grapes. These are the alveoli. When somebody has emphysema, it really decreases the surface area, and then they have a lot less area for that simple diffusion of oxygen and carbon dioxide. Osmosis is the net diffusion of water across a selectively permeable membrane. So water is a molecule just like any of these solutes, even though we may not always draw them in our images or we can't observe it necessarily. Water also diffuses down its concentration gradient, which means water is going to go from an area of its own high concentration to an area of its own lower concentration. Notice that means it goes from an area of low concentration of solute to an area of high concentration of solute. So that means that water is going to follow the concentration of solute. And what this can do is it actually can change the shape of a cell or a membrane. So again, this is supposed to mimic a cell. There's a high concentration of solute in the cell, and water is going to go in. And you can imagine if a lot of water goes in, this cell, this membrane, is going to get bigger and bigger because it's filling up with water. This is going to occur until it reaches somewhat of an equilibrium, meaning the concentration of solute is equal on both sides. This is interesting to think about when we have osmotic pressure. So in this example here, you can see that inside this fake cell, we have high concentration. Water is going to go into it because water follows the solute concentration. Notice now, though, that there is this box around the cell. This box around the cell is going to impact how big this cell can get, right? And so in this, on the right side here, the box can actually tell us the pressure that the osmosis is pushing against this box. And so this pressure is made by water wanting to come in because the solute concentration is still high. Notice with the cell that has way more solute concentration in it, the pressure put on the box is higher. This is called osmotic pressure. A good example of this in real life would be plant cells and their cell walls. So cell walls kind of mimic this box that impacts, a cell isn't going to burst, for example, because they have this cell wall that impacts how large that it can get. The osmotic pressure is the force that would have to be exerted to stop osmosis. This is the force that's like preventing the more water from getting into that area. And it is proportional to the solute concentration, meaning the higher the solute concentration, as seen in that previous image, the higher the osmotic pressure. A clinical example of osmosis would be edema. Edema is tissue swelling, and this comes because there are a lot of plasma proteins in the area and water is drawn towards the solute concentration. Tonicity relates to osmosis, but it has to do with the effect that the solution has on the cell. So isotonic solutions have the same osmotic pressure. I'll show you an example. Hypertonic solutions have higher osmotic pressure. Hypotonic solutions have lower osmotic pressure. And let's see a clinical example. On red blood cells, an isotonic solution has the same osmotic pressure. And so what that means is that the movement of water in equals the movement of water out. A hypotonic solution has a lower osmotic pressure, which means that it will push water into the cell because it's following the solute concentration, and there's going to be more solute concentration within the cell than in this hypotonic solution. The opposite is true of a hypertonic solution. That means that there is more solute on the outside and the pressure is greater than plasma, and so water is going to want to come out. And what this does to a red blood cell shape is called crenate. It causes the cell to crenate or shrivel. And by the way, in a hypotonic solution, water flows into the cell until a red blood cell will pop. And this causes cell lysis. So back to that plant cell example, cell lysis wouldn't occur even though you put a hypotonic solution around a plant cell. Cell lysis is not going to occur because plant cells have that cell wall to maintain its shape and to impact osmotic pressure. This is super relevant to IV fluids. IV fluids have to be isotonic to blood. This is to maintain osmotic pressure and to maintain the shape and regularity of our red blood cells. It prevents our red blood cells from changing shape. If red blood cells were put in a hypotonic solution, they would lyse. And if it was put in a hypertonic solution, they would crenate. Mediated transport is a type of facilitated diffusion where, because it requires protein transporters, and this is to assist molecules that are too big or too polar to diffuse across the membrane themselves. So this can include some amino acids, the polar ones, and glucose. These protein carriers are proteins, and as you can remember from our last video, that means they have specificity. They are highly specific. They are only going to move one molecule. They may have competition where certain substrates may compete with the substrates for transport, and they may be saturated when all the carriers are occupied. When there is so much concentration of the substrate to be moved, the solute to be moved, that is when the rate of transport is at its highest, and so this is called the transport maximum. Remember that these carrier proteins are still a form of passive diffusion, and so it's still going down its concentration gradient, meaning from high concentration to low concentration. The image shows on the right here is a vesicle carrying carrier proteins to be distributed onto the cell membrane when more solute is required. This figure shows the concentrations of certain ions inside the cell and outside the cell. And what you can see is there are some really big differences here. There's way more sodium on the outside, way more potassium on the inside, way more chloride on the outside, and way more calcium on the outside. And so what this tells us is that passive diffusion is not the only way, because if it were, it would be trying to reach more of an equilibrium. However, we are going to get into active transport next week, and active transport is going to require energy, and so it moves solutes against their concentration gradient.